the immune system. That's what we'll start to discuss in this lecture video. We'll look at the innate or non-specific immune defense. And then in the next video, we'll take a look at our specific defenses. So innate immunity is essentially what we are born with. These are the non-specific defenses, the physical, chemical, and cellular defenses that the body has in place when we are born. Now, the one thing to note, as we're going to cover these separately in separate sections of the chapter, but they all work together. It takes all of these different types of defenses working together in order to be able to defend us against attack. If we think about all of the times that we have acquired some type of infectious illness, there's 10 times that we haven't because of these defenses that we're born with, as well as those that are specific that we'll get to in the next chapter. So the physical defenses that we're going to look at here are the most basic form of non-specific defense. So we have physical barriers, mechanical defenses, and then our microbiome. So if we look at physical barriers, we're talking about basically cellular junctions. These are junctions that bind cells together so tightly that invaders can't get through. And as you've seen, as we've gone through some of the virulence factors and the pathogens, some of them are able to secrete enzymes that can break down this cellular glue so that they can squeeze in between there. But as long as those stay intact, pathogens can't get through there. Of course, our most important, our largest physical barrier is the skin. When we look at the skin, we see the epidermis, the dermis, and the hypodermis. And, organisms would have to get through here. So as long as the skin stays intact, again, pathogens can't get through. Infections occur when that skin barrier is compromised or broken, in the case of a wound of some sort. The mucous membranes are those kind of what we consider wet membranes. So those that are open to the outside. So the mouth, the nose, the urinary and digestive tracts, and even the reproductive tract. Mucous membranes have cells that have tight junctions. So they're stuck together, essentially, if we look up here what a tight junction looks like. So the cells are stuck very closely together. They also secrete mucus. Mucus helps to trap debris and other particulate matter. And then a lot of these cells have cilia. And we have what's called the mucociliary escalator, which is very prominent in the the respiratory tract. Where the pathogens and other dust and debris get trapped in the mucus, and then the cilia move that up and back out of the body, and that's why we, cough or sneeze. In the digestive tract, of course, we know that there are a lot of illnesses that can affect the digestive tract. The mucous membrane that lines the digestive tract is a physical barrier. It also has goblet cells in there that help to secrete mucus and trap different substances. And then some of that material can be excreted in feces. If we look at the urogenital tract, there are epithelial cells called endothelia that line these. We also see this in the blood-brain barrier that we talked about in chapter 26. So these help to keep pathogens out as well. Then we look at mechanical defenses. Of course, that mucociliary escalator would be a mechanical defense. Excretion of feces with pathogens in it is, a mechanical defense. But also tears, the production of tears, that's a mechanical defense that helps to wash debris and bacteria from the eyes. Also, the flow of urine helps to, wash pathogens away and prevent them from getting into uh, the urethra, the bladder, the ureter, or the kidney. And so these would be physical, mechanical defenses, essentially washing the debris away. And then we have our own microbiome. We have resident microbiota. We have, in the vagina, there are a lot of pathogens, of course, the colon, the surface of the skin. So we have, and I've referenced these several times as we were going throughout our pathogens because they are so important to outcompete pathogens that may come in. So now going to some of our chemical defenses. So we have a lot of different chemical mediators that are able to inhibit microbes. Some of them are endogenous, meaning that they are produced by our own cells. Others are produced exogenously, meaning they are produced by microbes that are part of our microbiome. So if we look at some of these chemicals and enzymes, if we look at the skin, okay, so we've already looked at the skin as a physical barrier, but we have sebaceous glands or the oil glands that are secreting sebum that is released onto the skin surface through hair follicles. It's an endogenous mediator, basically just sealing off that hair follicle to try to prevent microbes from getting down in there. We also have um, exogenous mediators that are produced by um, different pathogens. Um, they can use lipase to break down that sebum, which produces oleic acid. This creates an acidic environment that doesn't allow other organisms to survive. Um, Low humidity, decreased sebum makes the skin less habitable for those microbes that do produce oleic acid, which can make the skin more susceptible to pathogens. So, it's important that our skin, doesn't dry out. We don't want to take away too much oil because that is helping to prevent some bacterial infection. Saliva contains enzymes, mucus contains enzyme, tears contain enzymes, and these all help to destroy pathogens. Urine is acidic, it has a low pH, and that helps to deter bacterial growth. In the female reproductive system, there is an exogenously produced substance called lactate that helps to inhibit microbial growth. This is produced by the lactobacilli, which are good members of the normal flora, and this helps to lower the pH and it helps to inhibit colonization, especially of candida, which causes yeast infections. In the respiratory tract, of course, we have mucus. We also have lysozyme and lactoferrin that are antibacterial in nature. Then we have a special group of cell-derived mediators called AMPs, or antimicrobial peptides. Some are produced routinely, so some are always produced, some are produced in greater quantities in the response to a pathogen. They can do different things. So here's just a few examples of these antimicrobial peptides, where they're found, and then what they do. So you can see that they're effective against different types of pathogens, and they have different modes of action depending on what they do. So these are produced by our cells. Again, they're very non-specific. They're just going to target whatever they might find. Then we have some proteins in plasma, which is the fluid portion of blood. We have complement, acute phase proteins, and cytokines. So acute phase proteins are antimicrobial mediators primarily produced in the liver. They get into the blood in response to inflammation. So basically, the body says, hey, we've got some inflammation here, and we'll talk about that process a little bit later. And that triggers the release of these. And they do different things. Some destroy bacteria, some are effective against different types of pathogens. And then this mannose binding lectin here activates the complement cascade. So the complement system is a cascade system that is more than 30 proteins. They're circulating in the blood as precursors, but then they become activated by something, including microbes. They're considered part of the innate non-specific immunity because they're always present. This allows them to be activated very quickly. And then this basically starts to activate these proteins that are going to eventually kill pathogens. They produce different outcomes: opsonization, inflammation, chemotaxis, and cytolysis. Opsonization is when the pathogen is coated by opsonin, and this basically allows our phagocytes, our macrophages, to find them and destroy them. And then inflammation just brings more of these amps and these cytokines to the area. Chemotaxis and cytolysis basically involved in cell death. And then cytokines are also proteins that are communicators. Cytokines can be used in this complement cascade. They can be used in apoptosis or cell death. So if a cell becomes infected, they can trigger the cell to essentially kill itself. It really depends on what type of function it has. And then that kind of dictates what they do. Three classes that we've really studied a lot are the interleukins, the chemokines, and the interferons. Interleukins are basically involved in modulating all of the parts of the immune system. Chemokines are going to recruit our white blood cells, which are basically our macrophages and our bacterial targeting cells, to the sites of infection. And then interferon are signaling molecules, and they are very important against viruses. We also have some inflammation eliciting mediators. One of these is histamine, which regulates and supports inflammation. Leukotrienes are also pro-inflammatory mediators. They are more potent and longer lasting than histamine. They can induce coughing, vomiting, and diarrhea, which can, of course, expel pathogens from the body. Cytokines can also stimulate the production of prostaglandins, which are basically going to cause fever. Fever will promote white blood cell activity as well as inhibit the growth of pathogens. Bradykinin is also another inflammatory mediator. And so here's a table that kind of summarizes those. Now, getting into our cellular defenses. So we looked at physical, we looked at chemical. Now we're going to look at cellular defenses. When we look at cellular defenses, we have to really kind of consider blood cells. We have red blood cells, which are erythrocytes. We have the thrombocytes or platelets that are involved in blood clotting. And then we have the white blood cells called leukocytes. So red blood cells, they carry oxygen, that's their role. Platelets involved in blood clotting, that's what they do. But the white blood cells participate in immunity. Some are going to participate in our innate immunity, and others we'll talk about in chapter 18 that are going to regulate our specific immunity. So I'm going to kind of skip through this hematopoiesis portion here and kind of jump down here to our white blood cells. So the white blood cells can be divided into two types, granulocytes and agranulocytes. The granulocytes are neutrophils, which are here with this multi-lobed nucleus. Eosinophils, which a lot of times they have these red granules in them, but they have a double nucleus here. And then the basophils have a very, very large nucleus, and then the granules that stain purple here. And so these are our granulocytes. Neutrophils are the most common type of white blood cell. About 70% of our white blood cells are neutrophils, and they are involved in the elimination and destruction of extracellular bacteria. So basically, they are floating around through the blood, but they can migrate out of the blood into areas where there is active bacterial infection, and they will kill the bacteria. So as neutrophils fight the infection, we'll get this accumulation of leukocytes, cellular debris, and bacteria. This is called pus or purulent discharge. When you see pus associated with the wound, that is a signal that your immune system is doing what it's supposed to be doing. The immune system has been activated. And so you have a little note here that historically, doctors believe that inducing pus formation would promote the healing of wounds. Of course, now we know that's not effective. And so, but it is a sign that the body is doing what it's supposed to be doing. Eosinophils protect against protozoan and parasitic worms. They also secrete histamine, so they do play a role in allergic reactions as well. Basophils are the rarest type. Eosinophils are the next rarest type. 1 to 2% of all of our cells are basophils. They are involved in the complement cascade. They can also promote inflammation. They also secrete histamine. We also have what are called mast cells, very similar to the basophils, but they leave the bloodstream and they actually go into tissues, blood vessels or nerves, and they can support inflammation there. Now, agranulocytes do not have those cytoplasmic granules. These are lymphocytes and monocytes. If we look at the lymphocytes, we have natural killer cells and B cells and T cells, which are involved in our specific defense. Monocytes differentiate into macrophages and dendritic cells. So natural killer cells or NK cells are lymphocytes that are nonspecific. And what they do is they recognize and destroy cells that are abnormal. So cancer cells and virus infected cells are going to be targeted by these NK cells. They recognize these MHC, these major histocompatibility complex molecules, on healthy cells, and they know to leave those alone. But if there are diminished or elimination of these MHC markers, the NK cell is like, hey, that cell is abnormal. I need to destroy it. And so these natural killer cells can seek those out and destroy them. So here's a healthy MHC1 or a healthy cell with the MHC1 molecule. The NK cell is not going to do anything to it. But this one, it's an abnormal MHC molecule, so it's activated and it will destroy that cell. Monocytes are the largest of the white blood cells, and they are phagocytes. Monocytes will leave the bloodstream and they will go into tissues and differentiate into macrophages and dendritic cells. Macrophage means big eater. So macrophages are phagocytes. They are going to look for cells that are damaged. They're going to look for pathogens that don't belong, and they're going to go in and ingest those. Dendritic cells are in the skin and the mucous membranes, and they're basically on the lookout for any invader that comes in there. Now, looking at pathogen recognition, so with talking about these phagocytes, so these cells that are going to essentially ingest and destroy other cells, it's important first to identify that they are infected with a pathogen or that they are a pathogen. And so in this section, we're going to look at how they're able to find and recognize and then destroy pathogens. So, like I mentioned a while ago, the leukocytes can leave the bloodstream. They have to get out of the bloodstream to get into the infected tissue. And leukocytes will stick to these adhesion molecules, and then they'll be able to basically squeeze in between those junctions between the cells and get out into the tissues. And this is called transendothelial migration. Now, doesn't occur in arteries and veins. It only is going to occur in the endothelial cells, which are really, when we talk about capillaries are only composed of endothelial cells. So it's very, they're very thin walled. And so here's kind of an example of those leukocytes being able to squeeze out and get to that site of invasion. And then in order to recognize these, opsonization of pathogens can take place where we surround that. Complement factors, lectins can all assist these phagocytic cells. There are also some other structures called PAMPs, pathogen-associated molecular patterns, like peptidoglycan or flagellin, the lipopolysaccharide. And so basically they allow our cells to recognize those as being foreign so that they know that they can seek them out to destroy them. And that, of course, is called phagocytosis. And I'm not going to talk about all of those. You can read through those steps on your own. Now getting into inflammation or the inflammatory response, of course, triggered by that cascade that we've been kind of discussing here. Inflammation, a lot of times gets a bad rap, but it is a necessary process because it is basically a recruitment process for all of those different defenses that we need. Now, excessive inflammation, of course, is bad, but we do want some inflammation. So acute inflammation is basically that immediate response to tissue injury. We get vasoconstriction of the blood vessels to help minimize blood loss. And so the amount of vasoconstriction is related to the amount of vascular injury. So, if it's a tiny little capillary, then the vasoconstriction would not last that long. If it's a larger area, then it could last longer. After vasoconstriction, we get vasodilation. This is due to the release of histamine. Basically, what's going to happen is the blood vessels are now going to dilate, and we get increased vascular permeability, which is going to allow those phagocytes to be able to get out of the bloodstream and into the area where they need to be. The increased blood flow also helps to dilute the toxins and bacterial products that are there at the site of infection. And we see 5 observable signs associated with inflammation: erythema or redness, edema, swelling, heat, pain, and altered function. So here is kind of our example. We've got mast cells that are in the skin. We get an injury, and we've got bacteria in here. So the mast cells are going to secrete histamine. And so histamine is going to cause our blood vessels here to dilate, okay? And it's going to cause that increased permeability. So the neutrophils are going to be able to get out of the blood vessel, out of the capillary here, into the area where they can start to ingest pathogens. And so the increased blood flow is going to make the area appear red because now the blood is closer to the surface. It's going to feel hot to the touch. Because our capillaries kind of become leaky here, this is going to cause the release of fluid also to the area, so we're going to get swelling. It's going to be painful because nerve endings are going to be triggered here. And then we could get a loss of function. Now, when acute inflammation is unable to clear the pathogen, then chronic inflammation may occur. And this can result in an ongoing battle between the host and the pathogen. At the superficial level, it looks like the wound has healed, but beneath the surface, this battle, this fight can still be ongoing. And chronic inflammation can also be involved in a lot of different conditions. Fever, part of the inflammatory response, of course, infects the entire body. It leads to an overall increase in body temperature. The hypothalamus is the part of the brain that regulates body temperature. But when we have certain bacterial or viral infections, this results in what we call the production of pyrogens. Pyro meaning fire here, or heat. So we're getting, and gen meaning generation of. So we're getting the generation of heat. These basically make their way to the hypothalamus. They affect that setting, and body temperature will become elevated. Fever enhances the innate immune defenses. It stimulates those leukocytes, like the neutrophils and those macrophages, those natural killer cells, to basically do their job. It also can inhibit the reproduction of bacterial pathogens. So that brings us to the end of chapter 17, our non-specific or our innate defenses. And then we'll take a look in the next video at our specific defenses.